All right, I'd like to introduce our next speaker, whose topic is bacteria, electricity, and you. Thank you. Thank you. A special person who I happen to personally know, with a fabulous sense of humor and an outstanding teacher. Steve Finkel is a professor in molecular and computational biology section of the Department of Biological Sciences at the University of Southern California. He currently serves as the U.S. Dornslife College Dean of Graduate and Professional Education and recently completed a term as the president of the American Society for Microbiology from 2020 to 2023. Research in his laboratory focuses on the mechanisms of long-term survival and the evolution of bacteria and the bioelectric properties of bacteria in laboratory and natural environments, including the human gut and the oceanic subsurface. Experimental approaches include studying evolving bacterial populations in real time, whole genome resequencings of evolving cultural populations, and studying the effect of environment on metabolic adaptation, including the ability of human-associated microbes to produce electricity. Now that's a mouthful, isn't it? So Steve, help us understand everything about you and actually your introduction. Thank you.
Thank you for that, Sharon. I forgot I gave you the non-layperson introduction. So if there's one take-home message from today's talk I would like you to leave with, and your friends and relatives are not going to believe you, it's that your poop makes electricity. I will show you evidence. I will show you the data. It is not something we expected, but it is the truth as I know it. So I'm going to share that with you today. So thank you very much for the invitation to speak. This is actually my second time speaking to Hazak. I did not have gray hair the last time I spoke to this group. I think it was in 2004. And VBS holds a special place in my heart, which you'll understand better in a moment. So before I start the talk, I'd like to encourage people to interrupt me. I'm going to treat this like we were in one of my classes, and when I give a lecture class, even with 200 students, I like to get interrupted. So if I don't finish the talk, that's fine. I can come back later. But I'd rather answer your questions. So you can either shout them out, or Joel might grab the mic if you want, and just raise your hand, and Joel will hand you the mic, and you can ask a question. So even if we're on slide 3, if you have a question by slide 3, just interrupt me. I'd rather answer your question then than save it for the end, because we may never get to the end. In my business, there never is an end. If I do my job, I'll leave you with more questions, like why does my poop make electricity? So what I'm going to do is first, in order to really help you appreciate what I want to talk to you about today, I have to start with a very brief history of the universe and the Earth. And I can do that in about 3 slides. Because it's really about understanding microbes, and in this case, bacteria. I'm a bacteriologist by training, and that's mostly what we study in my lab. And bacteria are everywhere, and they were here billions of years before human life was on this planet, and they will be here billions of years after human life is on this planet, whatever happens to the Earth going forward. And so I'm going to show you some evidence about that. But you know, when we think about bacteria, we frequently think about infection, and obviously they're bad bacteria. But the truth is most bacteria are good or benign, and you can't live without them. So I want to give you an introduction to that to really help you understand the context, and so we can get to the real, I don't want to use the word meat of the talk, because it's poop. So we'll get there. So that's the bioelectricity part. And then I don't think there'll be time at the end, but if there's time at the end, a few years ago I had the pleasure of working with some scientists at the Jet Propulsion Laboratory, thinking about how to identify bacterial life on Mars. So if there's time, we'll talk about that. That's a different part of my lab. But first, one of the reasons why I do feel a strong connection to VBS, there's a picture up at the top of my wife and I, now wife, Beth Rabin, the daughter of Al and Laura Rabin, if any of you know Al and Laura. There with Rabbi Schulweis and our ketubah when we got married in 1992. So we were married here at VBS, and I just checked with Beth. We had our hors d'oeuvres in this room. So 1992. So we could do the math on that. So that's a very important connection for me to this community. Okay, so let's talk about bacteria and people. So a lot of the work that we do in laboratory science is focused on humans, and there's very good reasons for that, and billions of your tax dollars are spent every year to help fund research studying everything there is about the human organism. But you can't really understand everything if you just study the human. You really have to study the environment, and you have to study other organisms in those environments. And for me, that other set of organisms are the bacteria. And so what's the connection between us and the bacteria? Why do I get not billions of your tax dollars, but at least hundreds of thousands a year to study microbes? And the connection is pretty straightforward. You know, bacteria are implicated in virtually all aspects of human biology. And disease, of course, when you get an infection, we want to cure that infection, and antibiotics, for now, are one of the ways we get to do that. But what we frequently don't talk about or think about is that bacteria are fundamentally important to just the normal functioning of the human body. So a lot of our research is allied towards understanding what the microbes are doing, and then if it happens to benefit humankind, well, yeah, that's a bonus. Okay, I'm a basic scientist, okay? Among the things that allows us to study bacteria and still make important insights into human biology is that bacteria really do, quote unquote, make a living the same way humans do. So the first thing is the chemistry of life is the same. So if you give a human some sugar and you give an E. coli bacterium some sugar, they're going to eat that sugar pretty much the same way with the same waste products. So it's the good stuff from eating sugar, and if you eat too much, it's not so great for humans. We've published work on essentially giving E. coli diabetes. If you give the bacteria a little bit, they love it. You give it a little bit more, they love it even more. You give it a little more, and they get sick. And the chemistry of what happens when those bacteria get sick is the same as the chemistry that happens inside humans. The difference is I can put as many bacteria as I want to in a test tube, and I can kill them, and nobody cares. So, you know, putting people in test tube, not a great idea, even undergraduates. But can't experiment on the undergraduates, completely forbidden. Mostly a good idea. Really? No, that one? Okay, I got one of my jokes landed. Okay, great. Note to self, don't experiment. It's your grandchildren, I know. The other thing that's really wonderful is there's this thing that we refer to as the unity of the genetic code. So many of you have probably heard of DNA, and we know that DNA is the biological blueprint of life. The way an E. coli looks at DNA and the way a human's cells look at DNA is exactly the same. They use the same code book to decode everything. They use the same machinery at the biochemical level. You can actually take some proteins from humans and put them in E. coli and they work. It doesn't work the other way. But the way this code book of life is decoded is the same for all organisms that are currently living on our planet. So we can understand a lot of how the biology of the world works, including the biology of humans, if we can understand in bacteria. And as I said, there's no lobby right now for bacterial rights. And you know, some of my best friends are humans, so it's okay. But you know, we spend a lot of time thinking about the microbes. And again, I'm a basic scientist primarily, so we're primarily in the laboratory. We do work with some samples that come from the field, but we're trying to understand the fundamentals of the biology of these cells. And if we can do that, then we can translate that knowledge to other systems, including people. So in order for me to get you up to speed on what I'm going to be talking about, I want to give you a very quick overview of microbiology. So I'm going to give you about a semester's worth of microbiology in about 6 minutes. You're all going to be experts. It's mostly pretty pictures. Well, I think they're pretty. You're going to go, what's that? But that's okay. And again, interrupt me if you have any questions. Okay. So how do we think about life on Earth? And there's lots of ways to think about it. This diagram represents what we refer to as the universal tree of life. And the words on it don't really matter. But what you see is a lot of branches and a lot of names. And I'll orient you in a second to where you fall on this tree. Every currently existing organism on Earth, that is, all the animals, all the plants, all the yeasts and the fungus, all the bacteria, everything that we currently know about falls somewhere on this tree. So this is how everybody is related. So we're all cousins, and we're actually cousins to the trees and to the, if you really want to take it to that level. The current theory is that all life on Earth that exists today is all descended from what is very likely a single species of microbe, going back about 3.8 billion years, which we call the last universal common ancestor, which I have here in red. So everything that's currently here, you, plants, animals, bacteria, fungi, are all descended from this thing that was like a bacteria billions of years ago. When we talk about how to categorize all this life, it falls, yes? That was a great question. So the question is, where do viruses fall? There's always one in every audience. So that is a fantastic question. And you caught me, because viruses are not on this tree. And the reason viruses aren't on this tree is because viruses are really hard and to study and to understand. And one of the questions becomes, are they living? Because a virus in and of itself is not a free-living, freestanding organism. It's an accessory biological entity which cannot reproduce unless it infects somebody on this tree and turns that somebody or something into a virus factory. So the viruses aren't on here. And what's more frustrating is the viruses can hop between all the branches. And we're hearing about that right now with bird flu. So you've touched on something fundamentally important. I am not a virologist, which some people will say with pride and others will say with trepidation. I'm a bacteriologist, and when I was the president of the ASM, I had a wonderful separate unit of virologists who took care of COVID stuff for me, which was fantastic. So viruses are fascinating and really important. And everyone has viruses. Bacteria have viruses, fungi have viruses, humans and plants have viruses. They don't show up on this tree. It would get, if we put the viruses on, it would be this giant interdigitated mess. It'd be fascinating. And actually, it's super fun to study. I like teaching about it. But this is a simplification. So thank you for bringing that up. It's a really important question. All right. So when we're trying to figure out all this life, taxonomy, the naming of things is really important in biology because we have to have a common language. And so I'm going to share with you a little bit of terminology just to help put things in boxes. So what I'm showing you is two boxes here, one called prokaryotes and one called eukaryotes. The names are not so important. The blue ones, everything in the prokaryote box, are organisms like bacteria. And those organisms are all unified by the fact that they don't have a nucleus, if you all remember going back to 7th grade science. And everyone in the green box is a eukaryote. They all have a nucleus. To help orient you, of all the life on Earth, almost all, there's always exceptions, but almost all the macroscopic life on Earth, right? I'm a microbiologist. I study microscopic things. Almost all the macroscopic organisms, that is, things you can see with the naked eye, only fit into this very small green box. And this includes the animals, the plants, and the fungi that you can see. Not all fungi are visible, but think mushrooms. And so of all the life that's represented by this tree, the only things you can see with the naked eye are in the green box. Everything else on Earth is microscopic, whether it's eukaryotic or prokaryotic. If you think about baker's yeast or brewer's yeast, you see the powder, but those powders represent billions of organisms. You can't see the individual yeast that you use to, bake bread or brew beer. They're all really tiny, but they fit in that same box. So here you are. So humans are here at the tip of the animals. But when you look at the totality of all the different kinds of organisms on the planet, we're really a very, very small tip of one of the branches. We're amongst the bigger organisms, and there's a lot of us, but all the macroscopic animals fit just in that little branch in terms of the genetic diversity of life that's on Earth. So the distance between these lines, all these branches, represents evolutionary time. The farther the distance, the more distant things have evolved over the history of Earth. So it actually, if you look at, for example, can you guys see my cursor okay? Okay. So here's the animals, and here are plants. And their distance on this projection is, that many units, right? If we go over here to the bacteria, an E. coli is over here in the proteobacteria, and, let's pick my other favorite, let's see, Bacillus subtilis, just another innocuous soil microbe, is over here. Their distance is much, much longer. But when I show you photographs of them, you can't tell them apart. They look the same. I'll show you some electron micrographs. These organisms can exchange genetic material. So they can essentially mate across billions of years of evolutionary time, which is one of the reasons why microbes can evolve so quickly. And then when the viruses come in, they carry genes around as well. Over here, these are all genetically isolated. Plants are plants, and dogs are dogs. Corn is corn, and dogs are dogs, and dogs mate with dogs, and corn mates with corn. But in terms of genetic distance, it actually makes far more sense for dogs and corn to mate because they're much more genetically related. We have far more in common with corn than we do with these microbes. And the joke I use in my class is, of course, what happens when dogs and corn mate? You get corn dogs, yeah, of course. That joke went over really big. I gave a talk at the Natural History Museum a decade ago, honey, something like that. Anyway, so it's the first time I ever gave a talk to a bona fide audience of drunk people. It was First Fridays, and the corn dog joke was huge. Okay, so I'm telling you that there's all this life on this planet, and it's been here for a long time. So let's just do a really quick history of life on Earth. So we have to start with the history of Earth itself. So the universe is around 14 billion years old. It's actually 13.75, but I rounded up. Okay, and then the Earth formed roughly 4,600 million years ago, or 4.6 billion years ago. So the Earth is about 4.6 billion years old. The early Earth was very hot. Liquid water was not possible for about 500 million years. And gradually, after the Earth formed, it began to cool, and finally it got cool enough that liquid water could exist. In our understanding of our kind of life, I'm not saying there aren't other kinds of life, but the kind of life we know about, you have to have liquid water. No liquid water, no life as we know it. So it wasn't until about 3.9 billion years ago that that's when liquid water first appeared, when the Earth got cool enough. So then what's really fascinating is relatively, quote unquote, quickly, if you are, a geopaleontologist or geomicrobiologist, the first prokaryotic life appeared sometime around 3.6 to 3.9 billion years ago. That number is probably more like 3.8 billion years right now. So that's our first evidence of prokaryotes, that is, things that look like bacteria. So Earth forms, we get liquid water, and within a few hundred million years, we have something that looks like the bacteria that we have today. Now, this is really fun to think about. When I ask my classes, and I'm going to ask you, where does the oxygen that we breathe come from? You're going to say, the air, the trees. It is fundamentally from water and through the process of photosynthesis. So a lot of times we think about photosynthesis has lots of products. One is it helps the plants get energy by taking the carbon dioxide from the air and fixing it into sugar that it can eat. And one of the byproducts is oxygen, molecular oxygen, O2, that we breathe. And that is absolutely true. But only about half of the oxygen we breathe on Earth comes from plants. The other half comes from bacteria that do photosynthesis the same way the plants do. In fact, the plants, quote unquote, learned how to do photosynthesis from bacteria. because the bacteria were doing it billions of years before we even had plants. And I'll show you that in a moment. So when the Earth first formed, when life first formed on Earth about 3.6 billion years ago, there were microbes, but they were not breathing oxygen. They were breathing other things, which is how we're going to get back to poop. Okay. Don't forget about the poop. All right. It took 1 billion years before there was actually oxygen in the atmosphere. And that's because around 2.6 billion years ago is our first evidence of there being actual molecular oxygen in the atmosphere. And the evidence for that is there were a lot of minerals on the Earth at that time, a lot of iron minerals. What happens when iron meets oxygen? You get oxidation, you get rust. And so in the geological record, as you go down, you go back through time, and there's all these rusty minerals, and then there's a point below which there is no rusty mineral. And that's because there was no oxygen in the atmosphere to rust anything. And that's because the microbes hadn't learned yet through the adaptive evolutionary processes, they hadn't figured out yet how to do photosynthesis. So there was no oxygen being pumped out in the atmosphere. So once those microbes started doing this, which for them was a really great idea, it probably caused mass extinctions because oxygen is actually kind of nasty. And it causes oxidation. I heard someone over here say oxidation. And it can cause damage to all sorts of biological molecules. So organisms also had to learn how to deal with that. Very nice lady in the back. Well, molecular oxygen. So the oxygen was always there, but it wasn't O2. It was tied up in other chemicals. So water, and then so H2O was there, and there was lots of other compounds that had O in them. They just weren't the O2 that's 20% of our air now. Because we breathe only, there's lots of oxygen in lots of things. We can't breathe water. We can't breathe the oxygen in water. But we can breathe the oxygen when it's just two oxygens glued together as a chemical. All the elements were all here. They just had to get rearranged by biology. It's an important question. Yes? Right, so the question is, how do they know? So these are always estimates, but they're probably good to 5 or 10%. So it's not off by thousands. You know, if it's 2.6 billion versus 2.6 million, that's off by 1,000-fold. But through carbon dating techniques and other techniques, a lot of it's done through the isotopes. So I don't want to go too technical, but carbon and nitrogen and oxygen, which are some of the main molecules of life, come in different flavors that we call isotopes. And different living organisms process those isotopes differently. So you can tell what happens when chemistry happens in the presence of life or in the absence of life, based on these isotopic signatures. And the geomicrobiologists have gotten really good at using various techniques, sometimes up to using synchrotrons and really giant atom smashers to pound these kinds of compounds so that we can actually figure out what they are made of down to the atomic level. So you have to believe in the physics and the math, which I'll just take their word for it. But they're pretty good at setting the clock these days. Yes?
I have a relative who is a micropaleontologist. Does that mean she studies fossils and things?
Yeah, if you're...
Repeat the question.
Yes, the question is, this person has a relative who's a micropaleontologist, so what do they do for a living? So they're probably applying these kinds of techniques to fossils. So you can then try to figure out, sometimes, what did that organism eat? So if it's dinosaur bones, for example, you can go look at the micro level and at the chemical level, use these kinds of isotopic techniques to see what they might have been eating, because different plants would have different signatures. There's also, there's DNA that's sometimes preserved along with the fossils themselves, or around them, you can begin to figure out what the environment is around those organisms. And start collecting all this information. So that they could very well be thinking about these kinds of things. These are great questions, by the way. Excellent. So we have life on Earth. We have oxygen in the air now for going back 2.6 billion years ago. After 2 billion years of having oxygen in the atmosphere, the first quote-unquote eukaryotes showed up. The eukaryotes are the organisms that look like you, for all intents and purposes. They were single cell in the beginning, but they start doing biology that eventually led to us as human organisms. And then it gets boring, because it's not about the microbes anymore. You guys care about the land plants 500 million years ago, and dinosaurs and mammals 200 million years ago. About 65 million years ago, a lot of the organisms, especially the big ones, the dinosaurs, had a bad day. And, current model is that a giant meteor hit the Earth and sent up so much dust into the atmosphere that it caused a major cataclysm, and the dinosaurs went extinct. And current models are it happened fast, like within days or weeks, the organisms all died. It was dark, and it got hot, and it was not good for the dinosaurs. But that left an opening in the biology of the Earth for other organisms, including little mammals who survived the cataclysm, probably because they were burrowing animals. And so little things like shrews and teeny tiny mice survived. And then, evolution picked up there. You get the first apes about 25 million years ago, and then you get the first organisms of the genus Homo, which is us. We're Homo sapiens, about 200,000 years ago. So I'm telling you this long history because I want to point out that over this huge amount of history, the microbes, what we call the prokaryotes, the first cells, they were here the whole time. So as long as there's been life on Earth, there's been life that looks like bacteria, and then we just came along much more recently, within hundreds of thousands of years. As humans, obviously, mammals have been around much, much longer. But the bacteria are always here. So now we have to appreciate, how many bacteria there are there and where are they. So let's play some numbers. So current events. Rough population of California right now, 40 million. 39 was what Wikipedia told me this morning. Or actually, whatever Google's AI bot said it was. But again, plus or minus. US rough population? Yeah, so about 340 million. And the rough world population. We just hit a milestone. Roughly 8 billion now. All right, so we've got an 8 followed by 9 zeros for the human population of the Earth, a very large number. I would be very happy to have that many pennies. Rabbi was talking earlier about someone who has way more than that many pennies. Guesses on the bacterial population, how many zeros? It's 5 times 10 to the how many zeros? 12, that's a good guess. Anyone want to go higher? To help bound your decision making, there's an estimated 10 to the 87 atoms in the universe. So that's 1 followed by 87, or 10 followed by 87 zeros. So it's somewhere between. Anyway, let's just jump to the punchline. It's probably a minimum of 5 times 10 to the 30th. So that's 5 with 30 zeros. So, again, if someone offers you that many pennies, take it. It's probably an underestimate. So this is an unfathomably large number. In my business, we have to simultaneously deal with 2 concepts. We deal with ridiculously large numbers of cells, right? Because there's a lot of organisms out there. But they're also incredibly tiny. So we have millions of billions of organisms we study, and they're a millionth of an inch or smaller. So it's a lot of what we call is the scale that we work on is very broad. All right, so let's try to get your head around 10 to the 30th of anything. If we took all the humans on Earth and laid them down, and this is a normal-sized human, so that's a Steve Finkel-sized human, I'm the tallest Finkel ever in my family. My in-laws and my wife will confirm this. At 5'7" according to my driver's license. You lay all the humans down end to end, head to toe, head to toe, head to toe. They would, 7 million miles. You do that with all the bacteria on Earth, and it's 20 quadrillion million miles. This is not helpful, okay? So let's think about it this way. You take all the humans and build a ladder out of them to the sun. Sun's about 92 million miles away. So those 7 million miles worth of people get you almost a tenth of the way to the sun. It's a reasonable distance. These bacteria, if we went back and forth between the Earth and the sun, you could do it 200 trillion times. So this is one way of saying there's a lot of these. Okay, and we have to, we'd like to know about all of them and give them names and understand what they do. How do you get so many bacteria? So in my laboratory, if you guys came into my lab and I taught you how to start some cultures, if we started the culture today and put some E. coli in a test tube with some E. coli food, the E. coli would double every 20 minutes. So human generation is 20 years, a bacteria generation in my lab is 20 minutes. So that means you have one now, in 20 minutes we have two, in 40 minutes we have four, in an hour we have 8. It goes up exponentially. It increases really quickly. If anyone ever says, I'll give you a penny and let's just double it every day for a month, take them up on it. Do the math when you go home. Okay, so if food supply was unlimited, that one cell, so today is Monday. If we came back to my lab on Wednesday, if we had unlimited food, it would weigh that many pounds, which is the equivalent of 3,500 Earths. So something that weighs a picogram, it's a 10 to the minus 12, so a trillionth of a gram right now, if we waited 2 days with unlimited food, would take over 3,500 Earths. They have prodigious reproductive capacity. The thing is, food's not unlimited. And so it's this interesting balance between the microbes wanting the food and what they can get their hands on, and how much can they grow, and then we have to keep them limited. So there's an interplay between us and the microbes, which brings me to my favorite. Yes, sir. That's a loaded question.
What was the question?
Thank you. The question is, how long do the bacteria live? This is a really good question, and it's something my lab studies a lot. So the answer is the following. We don't know, but like a good academic, one cell becomes 2 cells in 20 minutes. So are those cells still alive? Yes. How long did it live before it became 2? 20 minutes, okay. We have evidence that there may be places on Earth where the microbes can live thousands to potentially longer years without dividing. One of the places that we have samples is from about half a kilometer beneath the floor of the Atlantic Ocean. There are underground rivers under our oceans. And there are boreholes that go down into those underground rivers. So there's 10,000 to 12,000 feet of water, and then there's a well that's capped. And we either using remote operated vehicles or the manned submarine Alvin. I work with scientists who've gone down there, and they go to these wells and they drop bottles down, and they collect them every so often. And it looks like the waters are thousands of years old, and the microbes don't grow very much, but they're alive. So it could be thousands of years. And the way they do that is they may not be reproducing, but they're really good at doing repair. So we call it maintenance and repair. And they're really good at replacing their worn parts. So as long as they can replace their worn parts and they have just enough energy to keep all their biology going, we think they can live thousands of years. I have kept bacteria alive in my own lab without giving them food for more than 5 years. So we know it's at least 5 years, but we have evidence it could be thousands of years. Yes? Ask it again. Right, so the question is, if you're not reproducing, let me flip your question around. If you're not reproducing, then you can use that energy for other things. So reproducing could be a cost that makes you die faster. Completely rational model. Yeah, it's how it can be. And we really don't understand how these microbes are making a living. But we have a paper coming out soon, which will address exactly your question. So thank you for asking. Anything else right now? Yes. This is not a plant. I'm going to begin to address your question in 2 slides. So where can bacteria live on Earth? And the answer is anywhere that life can be, there are bacteria. So they're in virtually all aquatic systems, whether it's freshwater, saltwater, hypersaline water, the Salton Sea, the Dead Sea, or really, really pure water. There's microbes everywhere. Any dirt, forest, grassland, desert, anywhere there's dirt, there's bacteria. And they live both on the surface and beneath the surface of the planet. And actually, a lot of the microbes may be below the surface. And then, of course, associated with the animals and plants. So to begin to address your question, let's talk about how many bacteria there are associated with humans. So this is for a freshly laundered human. So this is fresh out of the shower. So your freshly laundered human has between 5,000 to 50,000 bacteria per square inch just on your skin, so like your forearm. That's right out of the shower. And this is good. These are your microbes. They're living in communion with you. They're helping to protect you. One of my dear colleagues at USC, Myron Goodman, gives a talk called, "How Come When I Sneeze, You Don't Die?" And it's a good question. And usually that's a talk about the immune system and how we can generate antibodies and protect ourselves. But actually your bacteria are your first line of defense. If my lovely spouse sneezes near me and those bacteria land on my arm, well, her bacteria land on my arm, but the Beth germs meet up with the Steve germs, and the Steve germs go, no, this is our human. We have dibs. And so the foreign microbes are going to have to work hard to overcome what I already have coming into the game. Then we have warmer, moister places. So the groin and axilla, that's the nice medical term for our underarms. 5 million bacteria per square inch. Then you get up to teeth, even freshly brushed. 5 million to 50 million per square inch of tooth. Many of you, I'm sure, have excellent oral hygiene practices. So for your average person who flosses, well, if you floss in the morning, well, what do you have to do the next morning? You have to floss. Why? Because it's bacterial. Dental plaque is alive. And it's the oral microbes are making all this stuff that you find in your mouth. And a lot of it's there to protect you and begin to help digest food. And then, of course, we're going to get to the main event is the colon. So here, it's 300 million bacteria per gram of feces. So a little pea-sized piece of fecal, I'm assuming everyone's changed at least one diaper in this room. A little piece of feces has, between 300 million and a billion bacteria in it. And then you can think about, I don't want to get too scatological, but I do study poop for a living. You figure about a normal bowel movement, and that's a lot of bacteria, okay? Every bowel movement's way more bacteria than there are people on Earth, just to give you some perspective. Can you imagine working in my lab? It's fun. It's really fun. So there's roughly 10 to the 13 bacteria per person. So that's 10 followed by 13 zeros. And now to begin to address the question that was asked earlier, there's roughly 10 to the 13 cells in a human body. So if we do it by sheer number, you are 50% human cells and you are 50% bacterial cells. You're half bacterial by number, not by weight. By weight, all your bacteria weigh about the same as your brain, so a few pounds. those 10 to the 13th microbes weigh a few pounds. So to your question about biomass, so you can figure out how many, multiply by 8 billion for how many humans there are, and so that's 10 to the 21 human cells, and then there's all the microbes associated with humans is 10 to the 21 as well, or 10 to the 22.
What organs have no bacteria?
The question is, what organs have no bacteria? This is a hot topic. When I was in graduate school, actually when I was in graduate school at UCLA and then as a postdoc at Harvard, I was in the medical school and we were frequently working with physicians and these kinds of questions came up. And I was actually taught that certain organ systems are sterile. And it is unclear to me now whether any organ system is sterile. We were taught the lower lung is sterile, otherwise you have pneumonia. We were taught uterus is sterile. We were taught lots of places are sterile, but it just doesn't make a lot of sense. So it's very possible that almost all organ systems have some form of a microbiota, and that microbiota is probably essential for helping to maintain the health and safety of that person. Molecular techniques have gotten sophisticated and sensitive enough that we can now find needles in haystacks. And there may not be a lot of microbes crossing the blood-brain barrier, but there probably are microbes in there. They're just not causing meningitis because they're good microbes. So potentially including the brain. This idea of gut-brain axis is a very active area of research. We know that many biologically active compounds are made in the gut, and those compounds make it to the brain. Jury's still out on for sure if there's microbes doing it in the brain, but the pathogens we know about, we know they can get there, and that's meningitis and other diseases. But there may be a natural microbiota in lots of places that were traditionally thought to be sterile. Urinary tract is another important one. There's really good data now, mostly focusing on women, that the female urogenital system has a native microbiota, and when that's healthy, the body is healthy. And that part of urinary tract infections is when other microbes can displace those healthy microbes. But traditionally, folks were taught urine is sterile. It's just that they couldn't catch it clean enough because there's a ways to go in the anatomy before you get to collect things. Excellent. Okay, so summarize the history of life on Earth. The first forms of life on Earth were almost assuredly prokaryotes, that is bacteria-like things. The oldest evidence from microbial fossils is about 3.8 billion years ago. And it is probable, it is almost certainly the way it worked, that all current life on Earth, extant life, is descended from these early prokaryotes, the early bacteria. There is a model that says Earth was inoculated from elsewhere. And the nearest elsewhere that is a candidate is Mars. That just pushes the question back of how life starts anyway. And that's a whole other, that's a course, not a lecture. There's an idea called panspermia, that meteorites, something smashed into Mars, which had life billions of years ago, sent meteors. The meteors had bacteria. The bacteria crashed on Earth and inoculated Earth like we inoculate our test tubes or Petri plates. You know, it's fine. It just pushes the problem back someplace else. So I'm going to show you a few pictures of what these microbes look like, and then we'll get to the poop. So this is my favorite bacterium. This is, question. Right. So the question is, what if we bring microbes back from other celestial bodies, including Mars? There is very serious concern about that. I don't worry about the Andromeda strain so much. Some of you may have read it or remember that movie, which is fun to think about. Well, not killing all the people, that's not so fun. The odds that a microbe from Mars would cause catastrophe on Earth is very low. Having said that, there is a facility up in the Bay Area which has never been used because we haven't brought anything back yet. And I call it the mother of all containment facilities. You know, it is a building in a building in a building in a building where if we ever do bring it back, that's where it goes to keep it contained. It's going to be a long time till we bring anything back. What I'm frankly more concerned about is humans have been crashing a lot of stuff into Mars for a really long time. And we're not going to have time to talk about it in any detail, but there are people with my favorite job title ever who I met at the Jet Propulsion Laboratory. Their title is Planetary Protection Officers. And their job is not to protect the Earth. They will eventually be the ones to protect the Earth. Their job is to protect other planets. And these are microbiologists who have tried their best to decontaminate every rover we have sent to Mars. And literally with Q-tips. Because there's actually a treaty that most of the countries of the world have signed, a UN treaty that says we will not send anything with terrestrial Earth microbes to other bodies, Venus and Mars and the asteroids. Not all countries are as assiduous about it as we are, but it's a real thing that people worry about. You know, the first person to go potty on Mars, you know, it's not like they're going to bring it back. So there's going to be a porta potty on Mars one day, right? It's too late for the moon. It's too late for everything. And frankly, I don't mean to cast aspersions, but, the Soviets weren't as good at decontaminating things as we were. But even we make mistakes sometimes. So I just want to give you a little tour of these microbes really quick. So these are, in the broadest sense, microbes come in two flavors: spheres, called a coccus, or the pluralis cocci, and rods, which are called bacillus. So this is Staphylococcus, so hopefully no one's ever had an infection with a Staphylococcus, but this is what staph looks like. These are Diplococci. These come in pairs. They're like twins that never quite want to get away from each other, so they just come in pairs. We call those Diplococci. These are Schizococci. These are really cool. The white things you see are actually essentially airbags inside the cells, and this is a submarine microbe. It can go up or down in the water by inflating or deflating these gas sacs. And so it's very smart, and it likes to find where it wants to be, and if it's too high, it lets gas out, and if it's too low, did I say it backwards? If it wants to go up, it inflates, and if it wants to come down, it deflates. Very clever. I really like these. These are called Sarcina. And they come, these are the first organisms I know of that can count. But they can only count, you have 10 fingers, so we count in base 10. These organisms count in base 2. They only come in 1s, 2s, 4s, and 8s. You never see 3s or 5s or 6s. So they know how to count, but they only know how to count in a sort of binary kind of way. And this is what's affectionately called the string of pearls. This is actually Streptococcus. This is what causes strep throat. So everyone in this room has probably at some point had a strep throat infection, and this is what it looked like at the microscopic level. Then you get your rods, your bacilli. So this is Bacillus subtilis, which I said looks just like E. coli. They're 2 billion years separated in evolution, but microscopically you couldn't tell them apart. These are curvy bacteria. This curve is called a Vibrio. That's the shape and the genus. This happens to be Vibrio cholera, the causative organism for the disease cholera, which is a really terrible diarrheal infection. To stay in the theme of things to make you slightly uncomfortable, these are corkscrew-shaped bacteria. These are called a spirochete. This happens to be Treponema. This is the causative agent of syphilis. Because, what's a little bit of sexually transmitted disease organisms between friends? This is a filamentous organism, and this is a cyanobacterial organism. This can actually do photosynthesis. So it grows like bacteria would normally grow, but it grows in these filaments. And the reason I want to share it with you is, if you look microscopically at these cells, you'll see 3 different cell types. And the cell labeled H is the one that does photosynthesis, and the cell labeled A is the one that's involved in the reproduction of the organism. And then the rest are called vegetative cells. So there's 3 cell types. So this is one string of bacteria that has compartmentalized itself. The reason I bring it up is the regular cells and the A cells and the H cells all have exactly the same DNA, but they do different biology. So the biology of the H cell is fundamentally different than the rest of the cells. And this is just the same as we have the same DNA in all of our cells. But we have kidneys and brains and liver and bone. And with the same blueprint, they're doing all these different activities. That's not unique to multicellular organisms like mammals. It's also specific to bacteria, which are far simpler. So this idea of division of labor goes back billions of years. And then the exceptions proving the rule. I told you bacteria are microscopic. You can't see them with the naked eye, except for the ones that you can. So this is the second largest bacterium in the world. So there's 1, 2, 3 cells here of a microbe called Thiomargarita namibiensis, sulfurous pearl of Namibia. Came out of a sulfurous, rather stinky lake in Namibia. This is a fruit fly. So you can see here's the fruit fly's head, and here's one of those cells. So there's bacteria that actually are macroscopic. It's actually now the second largest bacterium. This is the current winner, is a different Thiomargarita called magnifica. This came out of a mangrove swamp in Guadeloupe. So there's some macroscopic microbes, which is an oxymoron, but it's just what we have to deal with. Okay, finally, the last few minutes. Your poop makes electricity. Okay. So what I'm going to introduce you to is some work that my lab does studying microbes that come from the human gut, that we're amongst the first labs to discover that they have this amazing property that they make electricity. And the process is called extracellular electron transfer. And it's really just another way for bacteria to breathe. Breathing is fundamentally about getting rid of electrons. And I don't want to get into the chemistry in any detail, but all you need to know is that when we eat food, there's a lot of byproducts. And amongst the byproducts are excess electrons. And we have to get rid of them. Otherwise, our metabolism stops. And the way humans deal with it is we dump those electrons onto oxygen. That's why you have to breathe air. Microbes do it exactly the same way we do. They breathe in O2 and they breathe out CO2. The difference is if you put a bag over my head, I'm not going to last very long. If you put the equivalent bag over bacteria, they'll just find something else to breathe. Because remember, when they were quote unquote born, when they first evolved billions of years ago, there was no oxygen to breathe. So they already know how to do it. So they can live in places with air and places without air. And one of the places without air is our gut. There's no free oxygen in the human gut. So this is instead of breathing oxygen. So I already told you that part, good. So here's a few bacteria. And what happens is, if they're in the presence of oxygen, then the oxygen can diffuse to those cells and the electrons will be taken away. But what's really cool is there's a subclass of microbes that can take those electrons and transfer them to something else to breathe. And in this case, those electrons could go through this extension down to something else. These microbes were first discovered in Lake Oneida in upstate New York, in one of the Finger Lakes, breathing rock, which doesn't make any sense. Breathing rock. And what they were doing was they were taking a mineral that was a rock, and they were donating their electrons to that rock, and the rocks were dissolving. And one of my colleagues at USC discovered this, a scientist named Ken Nielsen, and he said, hey, I think they're breathing the rocks, and everyone said, you're crazy. And he wasn't crazy. Well, he's a little crazy, but he happened to be right. A better analogy is if you take a rusty nail and you put it in a test tube with this bacterium, the nail will stay rusty forever. If you put a cork in that tube, so no air is getting into that test tube, in a day, the nail is clean. All the rust has been breathed by those microbes. So this is what we were studying. We were actually making batteries with bacteria that could transfer these electrons around, and we were making electricity. It was never going to run your Prius, but it was fun. We put in some sugar. We could make a light bulb go off. It was really cool. And then that program got canceled. And the folks at the Air Force who was funding this research said, hey, Steve, do you think this is happening inside people? Because they had a real problem with travelers' diarrhea when they deploy airmen. And no one really understands why that happens. And they said, we need to understand more about the human gut. And they said, maybe, go see if this is happening in people. And they gave us a little bit of a grant money to look at this. And sure enough, we discovered it's happening inside people. So we think this kind of electron transfer is happening associated with many parts of the human organism: gut, in the mouth, the lung, nasopharynx, that's up your nose. There's certain wounds that we think are electroactive, and in the urogenital system. But I'm going to focus on the gut. So the human gut is really just a big bioreactor. Food goes in one end, feces comes out the other, and there are many, many different kinds of bacteria along the way. And all these bacteria have to breathe, they have to do biology, they have to survive. And so what we're doing now is we're going into human samples and asking what kind of biology are they doing. Okay, so how do we actually study this? How can I convince you that your poop makes electricity? So we have a device called a bioelectrochemical system. It's essentially a jar. There's no oxygen inside. It's got a lot of electrodes in it. It's run by a computer. And we introduce bacteria into this chamber. And if they're making electricity, they will touch these electrodes and we can detect those electrons. And we can detect the electricity. What we initially did was we got a sample of bacteria that came from 47 healthy humans. And it came in a little test tube. And we put it in our device. And what happened was, after we added the sample, after a couple hours, we started seeing this electricity being generated. And we've now done this many, many times. And all the samples we study have microbes that can make electricity. But now what we have to understand is who is actually doing it. We have many species that can do it. Here's just a few of them: E. coli, Klebsiella, and Pterococcus. What's really, really cool, and something that we're very excited about, is the microbes aren't simply making electricity, but we also know they're talking to each other in the test tube. And if we get the right pairs together, they actually make more electricity than they would on their own, which is helping us to understand why would they even do this in the first place. It may be that some are taking the electrons and some are giving the electrons. And the way we can show that is the following. This is two of my species together. Here's an E. coli. Alone. Here's E. coli by itself, and here's something called Enterococcus faecalis. You can tell from the name where it came from. If we mix them together, we don't get any increase in electricity. But if we grow them together, in these experiments we grew them separately and then we put them together. But instead, if we grow them together outside of the device first and let them get to know each other and talk to each other, and then we put them into the device, you don't need to have a PhD to know that this curve is a lot bigger than this curve. And we actually see 400% increase in electricity production. So this is telling us the microbes are talking to each other in the test tube. And what we really want to know is, if they're talking in the test tube, are they talking in the human gut? So this required us to collect fresh fecal samples. So this is something that we do in my lab, big fun. And we get some highly trained volunteers, and we'll call them A, B, and C. These are anonymous donors, otherwise healthy, male and female. And we got their bacteria. And the important thing is we've now done this 5 times. Every single time, it's within 10 minutes of producing the sample to getting it into our device in the laboratory. And the minute we add a little bit of their poop, we see electricity. So this is why I'm convinced it's happening inside everybody all the time. You're not going to run your Prius on it, okay? But it is happening. And so what we're trying to understand now is, what are all these bacteria doing? How are they talking to each other? Who are they and who are they talking to? So we go back into the samples. Here's a bunch of bacteria that came out of people's poop. And here's the important part. We have in this one sample all these microbes. And now what we're doing is every possible combination of the folks in red are my current team of graduate students and collaborators at USC. I've been really blessed to have fantastic PhD students and postdoctoral fellows over the last few years. And I've also been very blessed to have funding from a variety of agencies, from the NIH, the National Science Foundation, some of the defense organizations as well in the US. Question? Well, the end goal for us to understand it, or for the microbes? Well, what we can expect is, we think when the human gut doesn't make electricity properly, you may get what we'd call a dysbiosis. You may get diseases, maybe IBS, maybe Crohn's disease. Maybe those diseases that we think of as autoimmune aren't actually just about the immune system going awry. It may be about an imbalance of the microbes in the gut, also an imbalance of those microbes talking to the human cells. Something that scientists really want to understand is, are those electrons also talking to the human cells of the gut? Because the way neural signals are transmitted is it's through electricity. It's a different kind of electricity, but neural transmission is also based on electricity. And so one of our ideas is that the electricity being generated by the gut organisms might also be talking to the humans, and this back and forth is required for proper health. So that's one possible outcome of this work. Well, we used to. We made poop batteries, but they don't make that much electricity. But, and then we'll get to the last question really quickly. There are applications for poop batteries that are happening now in wastewater treatment. And we have a former PhD student who started a company based in San Diego where she has a gizmo about the size of a porta potty, and they use, it's farm waste, like pig slop in ponds, and that's fuel. And there's enough food in there to feed the microbes to make almost enough electricity to run the pumps. And we put a solar panel on the roof and it's enough. And what comes out are two byproducts, electrons and clean water. So this is actually, and my favorite application at a Foster's brewery in Australia. Bad beer, microbes ate it, made the electrons, ran the pumps, clean water came out the other side. Last question in the back. Yeah, that's a great question. Probiotics. What about probiotics? I am convinced that for some people, eating some microbes gives relief or helps keep a good gut. The problem is the science is not there yet where we can figure out which probiotic we should give a given person. So there might be something out there that works for you, but it gives the next person diarrhea and the person after that constipation. And there's an anecdote I tell about my own mother who found a yogurt at Trader Joe's that made her feel great. And she was dosing herself, half a yogurt every other day, and things were like clockwork. And she recommends it to a friend, and it was not the same outcome. And on that note, I guess I better stop. And thank you all for your questions. I really appreciate it.
Thank you so much. Very interesting. Just